So G1 cyclins essentially activate S cyclins. What do S cyclin CDKs do? That's a little bit more complicated, but it can be simplified. Here's a diagram from your text, 1718. It has a lot of words that I don't want you to pay any attention to, really. But let's just talk about what is involved in an S phase, the synthesis phase. It's the synthesis of DNA. So we're going to go from one copy of the genome to two copies, ultimately, in S. So how is it that a cyclin CDK can accomplish that, basically, through kinase reactions? And the answer comes from the way the replication proceeds. It begins with the recognition of a particular region, or many particular regions on the DNA that serve as the origins of replication. So DNA polymerase is going to bind here and start synthesizing DNA in both directions with replication forks and Okazaki fragments and all that stuff from molecular cell biology that I'm not going to go into. But that process has to be initiated so that DNA polymerase knows where to start, but because it does not have that information built in. So that's achieved by something that's called an ORC, an origin recognition complex. And I want you to look at this complex really only with the thought of where is phosphate and where is it not. So in the initial stages of the replication initiation, ORC binds, and then there is this accumulation of proteins that bind to the DNA as a result of the fact that ORC bound. And that leads to a series of steps that I don't want you to focus on too much, but notice that what's going on here is that we have the final accumulation of components that produce replicases, the DNA polymerase that can now actually move the replication force and make a copy of the genome, and everything is phosphorylated. So this is what the S-phase cyclin CDK has done. It has phosphorylated the components, first of all, of the ORC, but also individual components of the replicase. So that phosphorylation is required for those activities to kick in. It's not required of ORC to bind to DNA, right, because it's done that up here. But what that phosphorylation does is prevent the subsequent accumulation of components of the replicase from accumulating again. Because if all of those proteins bind to this ORC, then you'll get the synthesis of 4 copies of the genome when you only want 2, or 8, or whatever. So you do not want multiple firings of this origin of replication. You want exactly one event per cell cycle in order to prevent the accumulation of multiple copies of the genome. So it's that phosphorylation that regulates the initiation of replication with, once again, a cock and fire mechanism in which you have to cock the origin of replication before it fires, and then you won't fire again until you've cocked it again. And that cocking is the phosphorylation and then subsequent dephosphorylation of the ORC, which makes it possible to do the next round of replication in the next S phase. So here you have an unphosphorylated ORC. Why is it unphosphorylated? And the reason is the same one that we talked about in the case of RB. There is that period in the cell cycle where kinase activity is low or negligible, and phosphatase activity continues. So that will remove the phosphate groups from the ORC, and that sets the stage for the next round of synthesis that's going to occur here during this S cyclin activation, if it occurs at all. It may never occur if the cell goes into permanent arrest, which sometimes happens. And then finally, what goes on here in this green period? What did the M phase cyclin CDKs do? They do a bunch, and a lot of that I'm just going to leave out altogether. But some of the things that they do, that is how kinase activity can control progression through something like mitosis, a very complicated cytological process. Some of that is shown on these diagrams from your text. And before going into that, I just wanted to do a real simple indication of what's going on in the case of mitosis. So we'll do a fibroblast, not an epithelial cell this time. But what happens in going from an interphase cell to a mitotic phase cell is that the nuclear membrane looks as though it just disappears. It doesn't actually. It vesiculates. It becomes smaller spherical membrane-enclosed vesicles. But as that happens, the DNA, which is interspersed, and uncondensed in interphase, invisible through a light microscope, becomes visible because it condenses, and that you get the individual chromosomes, and they're actually pairs of sister chromatids. But in any case, they condense, and then they line up in the way that you've had to memorize, I'm sure, since probably elementary school, maybe high school. And that lining up makes sure that the appropriate number of sister chromatids go into the daughter cells during the steps preceding cytokinesis, blah, blah, blah. So we have to accomplish that with a kinase, basically, a cyclin, M-phase cyclin CDK. So how do you do that? And again, it's very complicated. This is only a part of it. But let's deal with the condensation of chromosomes first. That is actually catalyzed by a protein called condensin, or partially catalyzed by condensin. So one of the things that the M-phase cyclin CDKs do is phosphorylate and activate condensin, so that you go from interphase dispersed DNA molecules of the genome to those very, very tightly packed, condensed chromosomes that we associate with mitosis. So that's a real simple thing that an active M-phase cyclin CDK can get you. The rest is a lot more complicated. So we talked about the APC complex as an important E3 ligase for cyclin CDKs just in general terms. There's a slide in the previous video that covers this exact same thing, but here we are actually using it to do what it does, which is to get rid of the M-phase cyclin CDK, but also to make the separation of chromosomes happen in a logical, carefully choreographed way. So here is the active M-phase cyclin CDK that's going to activate through phosphorylation the E3 ligase that does many things, but among the things that it does is lead to the ubiquitination and therefore destruction by the UPS of a protein called cyclin. Cyclin is a protein that binds to, and when it's around, inactivates another enzyme called separase, and that should give you some indication of what's going to happen next. In the presence of active M-phase cyclin CDK, you get ultimately the inactive destroyed form of cyclin and the active form of separase, which then binds to the DNA while it's still attached at metaphase, lined up, and then degrades the protein that called cohesin that links the two sister chromatids. And as a result of that degradation, you now have the possibility of separating those individual daughter chromosomes by being pulled apart by the microtubule, stuff that you learned, I hope, in cell biology and molecular cell biology. So that process can't happen until you have the active M-phase cyclin CDK. And the overall timing of the events is very carefully arranged so that you don't get separation before you get alignment, because otherwise you would get two chromosomes going here and none going there, kind of thing. So this careful control of the timing of the M-phase cyclin CDK is one of the beauties of that system, the fact that you can control it through all of the mechanisms, including gene transcription, but also much more rapidly modulatable things like phosphorylation and proteolysis of inhibitors. All of that makes it possible to do these really carefully choreographed events, physiological events, modulated by kinase phosphatase activity. Okay, so to wrap up this section, I just want to talk a little bit about the sorts of mechanisms that would be associated with the cell cycle arrest. And they are often things that activate expression of those inhibitors of the cyclin CDKs, the little P21s and stuff like that, P27s. So this diagram, figure 1763 in your text, shows one mechanism, but I can use it to show another one as well. I've talked about DNA damage as a signal that triggers cell cycle arrest or apoptosis. We can now talk about almost specifically how that happens. I'll leave out some of the early steps, but ultimately what happens begins at this complex right here. So P53 is going to be a factor that, if left on its own, will activate inhibitors of the cyclin CDKs like P21 and or trigger apoptosis by mechanisms that we'll talk about in the next video. But it doesn't do that ordinarily if it's in complex with this protein MDM2, which is just an E3 ligase. It's just a very, very specialized E3 ligase that leads to the polyubiquitination and destruction of P53. So we talk about P53 as being unstable. It's not really, it's just that it's typically always very rapidly polyubiquitinated and therefore sent to the proteasome. So anything that leads to dissociation of this complex will lead to cell cycle arrest and or apoptosis, and DNA damage is one of those things. So it could be imagined as a pathway going in this direction on this diagram, if we modify it a little bit, because what happens is the phosphorylation of P53 that leads to the dissociation of P53 from MDM2. So that would look something like that. So now you have P53 in the freed from the chains of its controller, and once that happens, then it can go on and turn on the genes for cell cycle arrest, which is what we associate with, let's say, mild or correctable DNA damage. Or if that doesn't work, then the fail-safe mechanism is apoptosis. And we'll talk about that again in the next video. So what this diagram is actually talking about is a phenomenon associated with overproduction of a cell cycle stimulating transcription factor, MYC. So this is something that occurs accidentally and needs to be dealt with, but it also is something that we associate with a lot of cancers. Many cancer susceptibilities or mutations in the regulatory processes that are associated with MYC and that lead to too much MYC for our own good. So that's controlled at least partially through this pathway, in which we begin at the same place with a controlled form of P53. But one of the byproducts of MYC stimulation is the production of this ARF that is not the one of vesicular trafficking. Again, it's a different protein altogether. What it does is interact with MDM and essentially drains it away from its association with P53. So it sponges away the MDM, leaving you with free P53 that then activates the genes for cyclin CDK inhibitors or triggers apoptosis by mechanisms that we'll talk about next.